Good day, everyone. My name is Maxwell, and today we're going to be talking about the dental pulp under the following outline: introduction, organization, how this dental pulp is organized, its composition, the regions of the dental pulp, the functions, as well as the age-related changes. We're going to be doing this in the next 10 minutes maximum. So, by way of introduction, it is a loose connective tissue. Remember histology, we have different types of tissue. We have the dense, both regular and irregular dense connective tissue, elastic tissue. This is the a loose connective tissue. That is the dental pulp. It is formed from the dental papilla. I don't know if you can remember during a tooth development, we have the bud stage. Remember the cap stage. Remember something like this. You know that we have the bud stage, something like that, and then the cap stage where hold on, we have the cap stage, something like this, so we have the dental papilla right here, these dots. So the dental pulp develops from these region. Okay, so that's a little bit of the embryology. I said it's a loose connective tissue, and currently there has been no convincing evidence that we have elastic fibers in the dental pulp. Okay, so it could be, or it can be assumed more or less an inelastic tissue. Okay, it continues as the periodontal ligament at the apical constriction of the root canal. Okay, from this diagram, figure 1.1, this diagram here, you can see at this point, here, this is the root canal, right? And then we have the dental pulp coming downwards, and at the apical constriction, it continues as the periodontal ligament that attaches the tooth to the alveolar bone. Okay, so moving on, the dental pulp basically responds to stimuli, hence it is inverted. It is productive throughout life, with its major function being the production of the secondary dentin. It is contained within the pulp chamber. The pulp chamber is bounded, or its boundaries is the odontoblast. The odontoblast cells, which give rise to the dentin, or is responsible for the production of dentin. So, the organization, how is the dental pulp organized? It's organized into two major systems: the effector system or components, as well as the supportive system or components. For the effector system, it is majorly the odontoblast, right? Yes, that's the major effector system. That is like one of the most important, or it's the most important component in the dental pulp, right? Now, every other component or system, or rather every other component, makes up the supportive system. It supports the odontoblast. Either it protects it, it nourishes it, or helps it respond to stimuli in such a way, or that's the protection in a way. So it's every other cell, every other component, the blood vessels, the nerves, they are all the supportive components, hence the supportive system. So composition, the dental pulp is made up of fibers, non-fibrous matrix, the cells, the blood vessels, the nerves. So the fibers, we have the type 1 collagen, the type 3 collagen. We have the type 1 collagen being the most dominant, 60%, the type 3 collagen being 40%. However, we have type 4, we have type 5 present but very trace amounts. For the non-fibrous matrix, it is made up of the gags, the proteoglycans, and the proteoglycans. Now, for the made up of the gags, the proteoglycans, the cells, the blood vessels, as well as the nerves. So, for the non-fibrous matrix, the gags, that is the glycosaminoglycans, there are four major gags in the chondroitin sulfate, dermatan sulfate, heparan sulfate, and the hyaluronic acid. Now, the chondroitin sulfate in the mature tooth is 12%, while the hyaluronic acid is 60% in the mature tooth, meaning that the hyaluronic acid is more in the mature tooth, but it's the vice versa in a developing tooth and on or non-erupted tooth. Okay, being that the chondroitin sulfate is more than the hyaluronic acid in the non-erupted tooth. For the proteoglycans, you can understand what the proteoglycans mean. It's simply the glycosaminoglycans plus a protein core, a gag attached to a protein core. We have so many. We have the vesicant, responsible for production or creation of the gel, the syndecan, which contributes to the basement membrane of Schwann cells and endothelial cells. We have the biglycan, it regulates collagen fibrillogenesis, that is the formation of the collagen fibrils. The tennessine, responsible for cell movement, fibronectin, bind cells to the extracellular matrix, and the integrins, which act as the cell surface receptors. We also have the glycoproteins as another component of the non-fibrous matrix. Now, the glycoprotein, it's not in this lecture, I didn't write it down here, so just take note. It's simply your saccharides plus a protein core. The proteoglycan is your gags plus a protein core, while the glycoprotein is your saccharides plus a protein core. There are several, and they all make up the matrix. Now, as for the cells, we have the odontoblast cells, the fibroblast cells, the stem cells, and the defense cells. The odontoblast cells, they are post-mitotic. What does that mean? It means that it is terminally differentiated. Hence, if there is an injury to that cell, they don't have the ability, or yes, they don't have the ability to divide further, regenerating. So they don't have that ability. So injury to that cell can lead to the death of an odontoblast. However, thank God we have stem cells, which work or function is to replenish that store of odontoblasts in a way. Now, the odontoblast cells, they are columnar coronally, but in the root, they are more or less cuboidal. And they basically form a layer, a pseudo-stratified layer. I'm going to show you that in the next page. You can see here, you can see this layer where you have A, you can see the cells as we are. Yep, you can see a nucleus here, you know, in different up, down, giving it a false appearance as if it is stratified. So it's basically a pseudo-stratified layer. So coming back, I said it is post-mitotic, columnar, coronally, and radicular. In the radicular parts of the tooth, it is cuboidal. We have fibroblasts, cells also. We have the stem cells, which differentiate into odontoblast-like cells. And these odontoblast-like cells are responsible for the formation of reparative dentine, or tertiary dentine, or reactionary dentine. Now, when there is an insult to the dentine, that is damaged. Now, these stem cells give rise to odontoblasts. They are not odontoblasts per se, but they are odontoblast-like cells. Now, these odontoblast-like cells give rise to the tertiary dentine. We have the primary dentine, the secondary dentine, and the tertiary dentine. Primary dentine is what is there the bulk. Secondary dentin is formed in relation to age. The more the age, or the higher the age, or the older the person, the more the secondary dentin, occluding the pulp chamber. Okay, now, but for the tertiary dentin, it is only formed when there is an insult to the dentin, right? Yeah, so we have the stem cells, we have the defense cells, we have the T lymphocytes, the macrophages, and the dendritic antigen presenting cells, as the name implies, present antigen to our immune system and is responsible for the primary immune system. Right, so another component of the dental pulp is the blood vessel. We have blood vessels that get into the pulp through the apical foramen. Remember that we have the apical foramen right here, isn't it? So that's where they enter. We have the nerves that run side by side, forming the neurovascular bundle, side by side the blood vessels. Mind you, it's the pulp is highly innervated. About 2,500 axons enter through the apical foramen. It's also rich in blood supply. The nerves form a plexus just below the odontoblast layer, called the subodontoblastic layer or subodontoblastic plexus of Raschkow. Now, remember that the pulp or the dentin responds to stimuli, right? So if there is something there's some nerve terminals that actually run, as well as capillaries that run through the dentin. They pass the odontoblast layer into the predentin and then to the dentin. Okay, let me show you what I'm talking about. Somewhere here, let me use this as an example. Still using this, right? So we have this is our subodontoblastic layer around here, and then nerve terminals pass through to the predentin, even into the dentin. So impulses or stimuli can travel all the way down to this plexus where there is a reaction. These nerve terminals are made up of afferents of the trigeminal, as well as some autonomic, especially sympathetic fibers, right? The parasympathetic fibers, there's no major convincing evidence that we have it there. Vasodilation is basically due to the production of these peptides, like our neuropeptide Y, as well as release of nitrous oxide. All those help in vasodilation. So we have, like I said, it forms that plexus called the subodontoblastic plexus of Raschkow. Now, we have regions. Now, okay, this is the structure of your odontoblast cell. Structure of your odontoblast cell right here. Now, this is it. This is our cell. We have gap junctions all around. We have the nucleus right here. We have all these organelles, and then this is your process. You can see this nerve terminal, this green stuff going all the way with your processes, right, into the predentin as well as dentin area, forming, you see this whole forms the dentinal tubules, right? When we talk about the dentin in my next lecture, the dentinal tubules contains your process of your odontoblast, as well as your sensory axon and capillary blood vessel. So we have the regions. We have the supraodontoblast layer, the odontoblastic layer, and the subodontoblastic zone. Now, the supraodontoblast layer can be appreciated when we have a shrinking of the pulp. If I take a sample and due to the stains used or the process of the processing, we have a slight shrinkage, and this gives rise to a potential space. It's a potential space, actually. The supraodontoblastic layer is a potential space. Hence, it's not there all the time. So you can see the arrows there pointing to that. You can see this whitish, somewhat whitish line, right? So this is your supraodontoblastic layer, and this layer reveals some things. It reveals, number one, the predentin plexus of Bratlau. Remember that we have the below the odontoblast, we have the plexus of Raschkow. Now, above the odontoblast, but before the predentin, is the plexus of Bratlau, which also plays a role in sensation. Now, if there is no shrinkage to reveal this space, we can't appreciate this plexus. Another thing that is being revealed is the dendritic APC, that is the antigen-presenting cells. Of course, you know the function of antigen-presenting cells. Now, the odontoblastic layer, using this diagram, I'm sure you can appreciate the odontoblast here. These are all odontoblasts with the nuclei at various regions, forming a pseudo-stratified layer. So that has been appreciated. The last region is your subodontoblastic region, which is a cell-free zone. You can see where we have B. You can see how it looks a little bit whitish. It is anuclear and but contains some things, right? It is a cell-free zone of Weil, W-E-I-L. It's right here, cell-free zone of Weil, and it is, like I said, a nuclear, but contains your subodontoblastic capillary plexus and, of course, your plexus of Raschkow. Now, what is the function of your dentin? I told you majorly or primarily the dentin production during development, during adult life, as well as in response to insults. Remember, tertiary dentin. Now, what are the age-related changes accompanied with the pulp, the dental pulp? Now, the size reduces with age. Remember that we have the formation of a secondary dentin. As you grow older, the secondary dentin is being deposited, and it occupies it encroaches on the pulp, right? So let's use this diagram for example. We have the predentin, we have the dentin, right? So this is our odontoblast layer, is the subodontoblastic layer. Now you see this predentin over here. With time, it keeps on coming downwards, occupying this space. Very soon it will start. All here would be your predentin, leaving here as your pulp. So with age, the size of this pulp reduces, right? And finally, we have pulp stones, which is physiological, not pathological. In a radiograph, we would have calcification. Now, this calcification could either be snowstorm calcification or discrete pulp stones, which could occur singly or in groups. What do I mean? Pulp stones, if we have the stones scattered everywhere, that's your snowstorm calcification. But if you have pulp stones as one large big stone, or in groups, you have the stones in groups, that's now your discrete form of calcification. right? So these pulp stones, they resemble dental tubules. However, they are called false denticles, not true denticles. So ask you, your Viva, have you heard of false denticles? Is it pathological or physiological? You should know that talking about pulp stones, actually physiological because it's related to age. Yeah, so I believe we've come to the end of this lecture, the dental pulp. Uh, by now, you know how it is formed, where it is formed from. It's a loose connective tissue. Its function is simply dentin production. You know its age-related changes. You know the regions, the three regions. You know its components, and you know how it is organized. Thank you.